Hello, it's Shiden Nagar, and today we're going to be solving May-June 2024 Paper 1, Variant 1. And before we start, I'd highly appreciate it if you would take a moment out of your very busy day to subscribe to this channel and like this video. In addition, don't forget to check our website link in the description box down below, where you can access your free topic 1, 2, 3, and 4 notes. All you have to do is press on the link, log into an account, and press File Share to access your notes. Question number 1. The diagram shows a stage micrometer scale viewed with an eyepiece graticule using a magnification of times 200. Using the same magnification, a chloroplast is measured as 4 eyepiece graticule divisions long. How long is the chloroplast? Now, first of all, we have to start by identifying which one is the stage micrometer and which one is the eyepiece graticule. So, this is the stage micrometer scale, whilst this is the eyepiece graticule. Now, how to identify them? We must know that the stage micrometer is an accurately drawn scale of known dimensions engraved on a microscope slide, whilst the eyepiece graticule is a transparent scale, usually 100 divisions long, as you can see here, placed in a microscope eyepiece, and it has no scale. Now, what we have to do right now is we need to calibrate the eyepiece graticule on the stage micrometer. Now, how we do this is that we take the size of one eyepiece graticule, so this is the formula, size of one piece graticule unit, equals the size of the known micrometer division, so known micrometer division, divided by the number of eyepiece graticule units. Now, since we know the formula, let's give you the practical example of how to work this out. Now, first of all, since the answer is in micrometers, we are going to convert the 0.1 millimeters on the stage micrometer into micrometers. The way we do this is that we multiply 0.1 by 1000 to convert to micrometers, and this is going to give us 100 micrometers. Now, as we said, because we want to calibrate the eyepiece graticule, we want to use this formula here. Now, we know that 100 micrometers has 40 eyepiece graticule divisions. Therefore, to know the size of one eyepiece graticule, what we do is we take 100 on the stage micrometer, divide by 40, this is going to give us 2.5 for one eyepiece graticule unit. Now, because the chloroplast measures as 4 eyepiece graticule divisions long, we're going to multiply 2.5 by 4. And this is going to give us 10 micrometers, which equals to the answer A here. Therefore, the answer is going to be A. Question number 2, which range of cell diameters is typical for prokaryotic cells? So you should know this from your textbook. Prokaryotic cells range from 1 to 5 micrometers. Now the closest answer is going to be B. Question number 3, the diagram shows a typical animal cell. Where would nucleic acids be found? Now, as we all know, nucleic acid must be found in the nucleus, where the DNA is found, which counts as a nucleic acid, and the nucleic acid is also the mRNA, which leaves the nucleus and goes to the cytoplasm and then to the ribosomes on the rough endoplasmic reticulum, and you're going to also find nucleic acids on the ribosomes and in the chloroplasts and the mitochondria. But because this is an animal cell, then we're going to find them in the mitochondria, which is shown in 3. Now, where would nucleic acids be found? As we said, they're in the nucleus, as well as in the cytoplasm, as well as the ribosomes, as well as the mitochondria. Therefore, 1, 2, and 3, and the answer is going to be A. Question number 4, which structure is found in a typical bacterial cell? So the way to address this question is that you must know the composition of a bacterial cell, that it's composed of a circular DNA, which is naked in the cytoplasm. Now let's see the suggestions that we have here. A, introns. Now, first of all, you must know because this is a circular DNA, it doesn't have the normal mRNA expression, etc., etc. Therefore, introns is incorrect. Now for B, telomeres. As we all know, telomeres are found at the end of the chromosomes. This is to protect the DNA structure, so for protection. Therefore, we know that bacterial cells have a circular DNA, they do not have chromosomes, therefore telomeres are incorrect. C, template strand. So, because we know that bacterial cells have circular DNA, you know that replication must somehow take place. You know, it's not in detail in the curriculum, however, you know that some sort of DNA replication must take place, therefore there must be a template DNA strand, therefore this is correct. Now finally, D, capsid. We know that capsid is the protein structures found in viruses, therefore this is incorrect, and the answer is going to be C. Question number 5. What is the maximum number of hydrogen bonds that can form between a single water molecule and other water molecules? Now we have to define a few terminologies here. Electrons in a water molecule are held by covalent bonds, as we all know. However, the shared pair of electrons in a covalent bonds are not shared equally in the case of an H2O molecule. As we all know that oxygen atom has a partially negative charge, so a delta negative, or the symbol, and the hydrogen atom in H2O has a partially positive charge, or a delta positive. Now, a hydrogen bond is a weak bond formed by the attraction between a group with a small positive charge on a hydrogen atom and another group carrying a small negative charge. Now, let's explain this scientifically drawing a diagram. Now, as we all know, that oxygen has an outer shell of 6 electrons. Now, this forms a covalent bond between a hydrogen and another covalent bond between another hydrogen. Therefore, what happens here is that there are 2 electrons, 2 pairs of electrons on the outside which are not bound to anything. Therefore, this forms the partial negative charge of the oxygen, whilst that there is a partial positive charge due to the attraction on the hydrogen. Now, what happens here is that the hydrogen here, because of the partial positive charge, and there's a partial positive charge here, forms a hydrogen bond between an oxygen. And the same thing happens here. So this is the first hydrogen bond, and this is the second hydrogen bond due to the delta negative. And then if we look here, there must be another hydrogen bond formation. So this is the third one, and finally the fourth one. Therefore, in total, we have 4 hydrogen bonds forming between a single water molecule and other water molecules around it. Therefore, the answer is going to be 4, which is D. Question number 6. The diagram shows the structure of 4 amino acids in aqueous solution. Which 2 structures have an overall charge? So this question is actually way easier to answer than what you think. All you have to do is look at the charges here and see if they equal out. So in this case, negative and positive, so this does not have a charge. Here we have positive, positive, and negative. Therefore, there is a net positive charge, therefore this is charged. In alanine, they cancel out, therefore this is not charged. And aspartate finally has a net negative charge, therefore this is charged, because as you could see here, 2 negatives and 1 positive. Therefore, lysine and aspartate is charged. Therefore, the answer is going to be C. Question number 7. The table shows the number of carbon atoms and the number of carbon-to-carbon double bonds in the molecules of 3 triglycerides and the fatty acids they contain. Which description matches fatty acid 2 of the 56-4 triglyceride identified in the table as X? So first of all, you must know that a triglyceride consists of 3 fatty acids and glycerol. So bound looking this way. Therefore, you must earlier know that a glycerol molecule must contain already existing 3 carbon atoms. However, if you didn't know this information, it can be identified from the table. Now, first of all, you must identify the pattern. For example, 15 plus 18 plus another 18, it's going to give you 51. And now the other 3 carbon atoms are going to be for the glycerol, which forms the total triglyceride. Now, therefore, there is a pattern. Now, let's see the other pattern for the second option that we have here. Now, if we were to add 17 plus 18, this would give us 35. Now, 56, which is the total, minus 35 is going to give us 21. Now, we know that this consists also of the 3 carbon atoms of the glycerol, therefore we're going to subtract 3 from 21, and this would give us 18 carbon atoms. So now we know that we have 18, and therefore we know that we also have 2 double bonds because we're subtracting the double bonds. Therefore, we know if a fatty acid has a carbon-to-carbon double bond, this means that it's unsaturated. Therefore, it's going to be 18 carbon atoms and unsaturated. Therefore, the answer is going to be D. Question number 8. Collagen molecules are made up of 3 polypeptide chains interacting together. The individual polypeptide chain consists of a regular pattern of amino acids. Almost every 3rd amino acid is glycine. Which protein structures of collagen are described? So first of all, the 3 polypeptide chains, we know that if there is more than one polypeptide chain involved, this means that it's instantly a quaternary structure. And now, for the second thing that we see here is the pattern of amino acids. We know that instantly the pattern of amino acids is going to be the primary structure. Therefore, it's primary and quaternary, and the answer is going to be B. Question number 9. The masses of parts of hemoglobin are shown in the table. What is the mass of hemoglobin molecule? So you must know that a hemoglobin consists of 2 alpha globins plus 2 beta globins, and every one of these chains has a heme group. So this makes it 4 heme groups. So 15,126 times by 2 plus 15,868 times by another 2, because we have 2 alpha globins and 2 beta globins, plus 617 times by 4. This is going to give us 64.5 in kilo DA. Therefore, the answer is going to be D. Question number 10. What is the correct order of locations in the cell for the production of an extracellular enzyme? So we know that the production of the enzyme starts with a process called transcription in the nucleus. So in the nucleus, which there is an mRNA formed in the process called transcription. Then this mRNA leaves the nucleus and goes to the cytoplasm on top of a ribosome on the rough endoplasmic reticulum in a process called translation. So transcription in the nucleus, then translation in the rough endoplasmic reticulum, which has the ribosomes on top. Then for producing an extracellular enzyme in the modification, the protein needs to be further modified in the Golgi body. Therefore, the vesicles are released from the rough endoplasmic reticulum and go to the Golgi body. And finally, the Golgi vesicles are packaged, and these leave the cell by exocytosis as an extracellular enzyme. Now, let's see the suggestions that we have here. For A, we have nucleus, then ribosome, which what we said, nucleus happens the process called transcription, then translation on the ribosome on the rough endoplasmic reticulum, and then finally goes to the Golgi body to be modified. Therefore, this is correct, and the answer is going to be A. Question number 11. The graph shows the trend from an enzyme catalyzed reaction. Which labels are correct for the x-axis and the y-axis? Now, take it as a rule of thumb. Any moment you see a bell shape, you know instantly that the x-axis is either going to be the pH or the temperature. The reason for this is that because each enzyme has something called an optimum pH or an optimum temperature. Below this temperature, there's a low rate of reaction, and if you passed this temperature or pH, the rate of reaction also decreases because there's an optimum. And this only exists in the pH and in the temperature. However, if this were substrate concentration or enzyme concentration, the graph would look something like this and then end up in a steep line. So if this were the substrate concentration, the initial rate of enzyme catalyzed reaction would be increasing and increasing because there are more active sites being occupied until all the active sites are occupied. Therefore, there's a steep curve because the enzyme substrate reaction cannot increase further, because now the enzyme concentration here becomes a limiting factor because all the active sites are occupied. Therefore, the x-axis cannot be the substrate or the enzyme concentration. Now, let's see the suggestions that we have here. For A, the x-axis has the rate of reaction and the substrate concentration on the y-axis. This is instantly incorrect. Now for B, we explained why this is incorrect. Now for C, on the x-axis we have pH, and on the y-axis we have rate of reaction, therefore this is correct. And for D, this is incorrect, therefore the answer is going to be C. Question number 12, which statements about Michaels-Menten constants are correct? Now the definition of a Michaels-Menten constant is that is the concentration of substrate which we reach half Vmax, and it's the measure of the affinity of the enzyme. So as the Michaels-Menten constant increases, the lower the affinity, and as the Michaels-Menten constant decreases, the higher the affinity of the enzyme for the substrate. Now let's see the suggestions that we have here. 1, the higher the Km, the higher the enzyme efficiency. We said that this is incorrect. 2, the Michaels-Menten constant is a measure of the degree of enzyme affinity. This is correct. Thirdly, is defined as the substrate concentration at which the enzyme functions at half its Vmax or maximum rate. There's also correct. Therefore, the answer is going to be 2 and 3, which is D. Question number 13, the diagram shows how nicotine is transported from blood plasma into a cell using a type of co-transporter mechanism. Here we can see that protons move down their concentration gradient out of the cell, so down and out, whilst nicotine here is transported against its concentration gradient into the cell. Which statement correctly describes a similarity between co-transport of nicotine and the co-transport of sucrose? Now, let's briefly outline the process of co-transport of sucrose into the phloem sieve tube elements. So here we have a companion cell, and here we have a phloem sieve tube element. Now, what happens is that sucrose is found outside the companion cell, produced by mesophyll cells. Now, what happens is that a carrier protein actively pumps hydrogen ions out, so pumps hydrogen ions against concentration gradient outside the cell, and this needs ATP. And then what happens is that sucrose molecules are found outside. They combine with a hydrogen ion and move in back to the companion cell together through something called a co-transporter protein. Now what happens is that sucrose moves against concentration gradient inside the cell, whilst in this case hydrogen goes back in to the cell down its concentration gradient by normal facilitated diffusion using a co-transporter protein. Same here with the sucrose, by facilitated diffusion using a co-transporter protein. Now, let's look back into the nicotine. Now, here we have that protons move down their concentration gradient out the cell. Now, the comparison here that in a companion cell, protons actually move out against their concentration gradient, and this is an active process. And here, nicotine is transported against its concentration gradient into the cell. Now, this is similar in case of sucrose, sucrose goes back into the cell against its concentration gradient by normal facilitated diffusion. As we can see here, this is a process of facilitated diffusion. And another comparison, that both hydrogen ions and sucrose goes back inside in the same direction, so not in opposite directions as we could see here. So that's another difference. Now let's see the suggestions that we have here. A, the cotransporter protein generates a proton gradient by moving protons out of the cell by active transport. Now this is not the case here, as we could see that protons move down their concentration gradient outside the cell. And here for the sucrose, that protons go back in against concentration gradient. Therefore, there isn't a proton gradient. Therefore, this is incorrect. And the process of active transport happens when hydrogen ions here are pumped outside. Be careful, there are two steps here. First of all, the pumping of hydrogen ions outside, which does not exist here. And then the hydrogen and sucrose going back into the cell, which is shaped by this process here through a cotransporter protein. Therefore, this is totally incorrect. B, the protons are transported through the cotransporter proteins by facilitated diffusion. Now, this is totally correct. We just mentioned that they go back both by facilitated diffusion through a cotransporter protein. C, the protons move through a cotransporter protein in the opposite direction to the movement of nicotine and sucrose. Now look carefully here. It says that the protons move through the cotransporter in the opposite direction to the movement of nicotine and sucrose. Now this is incorrect. In nicotine, yes, it moves in the opposite direction, but I mentioned that in sucrose, they go back inside the cell, both the hydrogen ions and the sucrose in the same direction. Therefore, this is incorrect. D, the cotransporter proteins use energy from ATP to transport protons. Now this is totally incorrect. They go back in by facilitated diffusion, as we mentioned here, not by ATP. ATP is used to pump hydrogen ions and sucrose here out, but they're not used to co-transport these two back in. Therefore, this is incorrect, and the answer is going to be B. Question number 14. The diagram shows a simple metabolic pathway. Which statements correctly describe the enzymes taking part in this metabolic pathway? Now, let's see the suggestions that we have here. 1. They are globular proteins. Yes, all enzymes are globular proteins having different structures. Therefore, this is correct. 2. They all have the same tertiary structure. Now, this is incorrect. Them having the same tertiary structure means that every single enzyme that exists has the same shape of an active site. But here we have different types of substrate. Therefore, they're going to have different shapes. Therefore, different active sites. Therefore, different tertiary structures. Therefore, this is incorrect. 3. They all contain hydrogen atoms in their structure. This is correct. The reason for this is that they have beta pleated sheets and alpha helices in order to form the attraction in the active site between the substrate and the actual enzyme itself. Therefore, this is correct, and the answer is going to be 1 and 3, which is C. Question number 15. A student observed the effect of two different concentrations of salt solution on blood cells. Slide 1, no blood cells were visible. This means that they probably exploded or burst. 2. The red blood cells were visible but look slightly crinkled. This means that water left the red blood cells, leaving it crinkled. Now, which row correctly explains the results obtained? So, for the first one, it makes sense that too much water has entered the red blood cell down water potential gradient because the outside had a high water potential and the red blood cell had a lower water potential. This means that water went down concentration gradient inside the red blood cell. Now, in contrast to slide 2, this means that the red blood cell had a higher water potential, therefore water moved outside the red blood cell into the surroundings, therefore it was crinkled. Now, let's see the suggestions that we have here for A. Swelling of the cells caused them to burst. Yes, probably would have happened since the cells are not visible anymore. Now, for slide 2, the water potential of the cell was more negative than the water potential of the external solution. This is totally incorrect. For water to actually move outside the red blood cell, this means that the red blood cell actually had a higher water potential, therefore a less negative water potential than the external solution for water to move outside down water potential gradient. Therefore, this is incorrect for B. The water potential of the cell was more negative than the water potential of the external solution. Now, if the water potential of the cell was more negative, this means that the outside had a higher water potential. Therefore, this means that water went inside the red blood cell, causing it to burst. Therefore, this is correct. Now, for slide 2, the water potential of the external solution was more negative. Yes, this is correct, as we mentioned in the first point. For C, the water potential of the cell was less negative. Now, this is incorrect. If the water potential of the cell in 1 was less negative than the external solution, this means that the water would end up leaving the red blood cell, not going inside in slide 1. Therefore, this is incorrect. For D, the water potential of external solution was less negative than the water potential of the cell. So, this is totally incorrect. Because of the water potential of the external solution was less negative, this means that it had a lower water potential. This means that the water would go inside the red blood cell or outside the red blood cell. This means that the water would go outside, therefore causing it to be crinkled. Therefore, this is incorrect, and the answer is going to be B. Question number 16, which statement correctly describes facilitated diffusion? So facilitated diffusion is the movement of molecules down concentration gradient through a transporter protein. Therefore, it's facilitated. Now, let's see the suggestions that we have here. A, the process only occurs using channel proteins that change shape and that use energy provided by these cells. So this is incorrect. It occurs either by using channel proteins or carrier proteins, which either change shape or don't. Therefore, this is incorrect. Now for B, the process occurs using channel proteins or carrier proteins that may change shape. Yes, this is completely correct. C, the process occurs using channel proteins or carrier proteins that use energy. Now, facilitated diffusion is the movement of molecules down concentration gradient. Therefore, these channel or carrier proteins do not necessarily require ATP or energy at all. Therefore, this is incorrect. The process only occurs using carrier proteins that create a gradient to move ions in opposite directions. Now, this is incorrect. If it were to move ions in opposite directions against concentration gradient, this would mean that it's a pump which uses ATP. It's no longer passive facilitated diffusion. Therefore, the answer is going to be B. Question number 17, which row is correct for stem cells? So, first of all, can repair cells and can be involved in the formation of phagocytes. Now, first of all, stem cells cannot repair the actual cell. It repairs tissues by differentiating and maturing into different types of cells, but that does not mean that it could actually repair the actual cell. Therefore, this is incorrect. Now, can be involved in the formation of phagocytes. Now, this is correct because there's something called hematopoietic stem cells. These hematopoietic stem cells, they differentiate and mature into different lineages. So, in case of phagocytes, it's the myeloid lineage. Therefore, this is correct. And the answer is going to be D. Question number 18. Eukaryotic organisms grow and increase in size as a result of cell division. The diagram shows some stages of mitosis with one stage missing labeled X. What happens in stage X? So, here we can see two different stages. We know that mitosis is PMAT, prophase, metaphase, anaphase, and telophase. So we can clearly see that this must be the metaphase stage and this must be the anaphase stage. This is because in the metaphase stage, the chromosomes lie across the equator. However, in the anaphase stage, what actually happens is that now we have microtubule organizing centers at the two poles of the cell. So two centrioles at right angles form something called a centrosome. So the centrosome are the microtubule organizing centers, which form these spindles. Now these spindles attach to the chromosomes at something called the kinetochore or the centromere in the middle. Now, this occurs in the metaphase stage. However, in the anaphase, what happens now is that these microtubules are like ropes which start to shorten. Therefore, what happens is now the two sister chromatids are pulled and separated apart, which would give us this shape that we see here. The two sister chromatids are actually separated apart by the spindle. Now let's see the suggestions that we have here, and we know that this is anaphase stage. A. Spindle fibers attach to centromeres and the chromosomes line up along the equator. Now this is incorrect. Chromosomes line up along the equator and attach to the centromeres. As I said, this happens in the metaphase. Therefore, this is incorrect. B. The chromatin coils and condenses and the nuclear membrane disintegrates. Now, this happens in the prophase stage. Therefore, this is incorrect. For C, the chromosomes line up in pairs along the equator. This is also the metaphase stage. Therefore, this is incorrect, or early metaphase. Now for D, microtubules shorten so that the spindle fibers contract and centromeres divide. Yes, this is exactly what happens here in anaphase, therefore this is correct. Number 19, the graph shows various distance measurements taken from the start of metaphase of mitosis, which show correctly identifies the distance measurements for each graph. So for graph X, the distance shortens. So we're looking for something that shortens. And for graph Y, we're looking at the late stages for something that increases distance in the late stages. So this is at the start of metaphase of mitosis. Now let's see the suggestions that we have here. For example, A, distance between the poles of the spindle. So as we all know that the distance from the poles of the spindle are equal throughout everything because the microtubule organizing centers are at the centrosomes. Therefore, they share the equal distance. Be careful here. It's the distance between poles of the spindle. Therefore, this is incorrect. For B, distance between poles of the spindle, also incorrect. For C, distance of centromeres from pores of the spindle. So centromeres, as we all know, in the end of metaphase and the start of anaphase, they separate, leading back to our last question, they separate at the opposite poles. Therefore, the distance of centromeres from the pores actually decreases. Therefore, this is correct because they decrease because the spindle fiber pulls the centromeres towards the poles. Now, for Y, something increases here. So the distance between sister chromatids. So the distance between sister chromatids, as we see here, because they were attached to each other and now separated, it's very normal that the distance between them actually increase at the late metaphase and the start of anaphase. Therefore, this is correct, and the answer is going to be C. Question number 20, which row is correct for the start of anaphase of mitosis? So we explained anaphase twice in the last few questions. So anaphase happens when the microtubule organizing centers, also known as centrosomes, form the spindle. Now the spindle attaches to the sister chromatids, separating them apart. Now what's going to happen is that the two sister chromatids are going to be pulled apart to the opposite poles of the cell. Now let's see the suggestions that we have here, the form of DNA. So chromosomes separate the sister chromatids. As I just mentioned, anaphase, they are separated already. Therefore, they are going to be as separated sister chromatids. Now, DNA is associated with histone proteins. Yes, Be careful here. Take it as a rule of thumb. During mitosis in general, the DNA is always associated with histone proteins. Associated with histone proteins means that the DNA is tightly coiled together. You could see this at the start of prophase, for example. The reason for this is that DNA replication and transcription do not take place in this area in particular or in this time in particular. Therefore, it's tightly coiled. Therefore, DNA is always associated with proteins. Now, the state of cell surface membrane, as we said, the two sister chromatids are the only thing that's separated. The cell membrane is still intact. Therefore, the answer is going to be C. Question number 21, the diagram shows the chemical structure of 4 bases. Which diagrams show thymine and cytosine? So you must know that thymine and cytosine are pyrimidines. And pyrimidines have one small ring, whilst purines, which are adenine and guanine, have 2 rings and which are larger. Therefore, we instantly exclude 1 and 4. Now we have 3 and 2 is our answer. However, this is not required in the scope of the question, but if you were to find out which one is the actual thymine and which one is cytosine, how you would do this? So as we know that adenine binds to thymine via 2 hydrogen bonds and cytosine binds guanine via 3 hydrogen bonds. Therefore, here we could see 2 potential hydrogen bonds formation. Therefore, this is going to be the thymine. And here we could see 3 potential hydrogen bond formation. Therefore, this is going to be cytosine. Therefore, the answer is going to be C. Question number 22. What is the sequence of 4 amino acids in the polypeptide translated from the small section of the gene? So this is an actual transcribed strand of the gene. Therefore, this is DNA. Therefore, we want to convert this DNA to mRNA codons. Therefore, it's going to be GCC, CGG, GGC, and GCC. Now, GCC here we have, it's going to be alanine, and CGG is going to be arginine. Therefore, the answer is going to be B. Number 23, what does the process of translation require? So, in protein formation by the cell, this happens in 2 steps. Transcription, which is the mRNA formation inside the nucleus, and then finally translation, which is the translation of mRNA sequence on the ribosomes on the rough endoplasmic reticulum. Now, this process obviously requires either rough endoplasmic reticulum, which has ribosomes in it, and tRNA, and probably amino acids. Therefore, the answer is going to be D. Question number 24, the electron micrograph shows a longitudinal section of a part of the stem of a plant. What is the name of the structure labeled X? So probably the structure is going to be a phloem sieve tube element. The reason for this is that phloem sieve tube elements have a slight peripheral cytoplasm, as you can see here. And they have sieve plates, as you could clearly see the sieve plates, which consist of sieve pores. And this allows the mass flow of sap solution from one element to the next with little resistance through these sieve pores. Now, as we could see here, we could see the companion cells on the side. Therefore, we could clearly say that this must be a phloem sieve tube elements, because if this were to be the xylem are hollow. They do not have sieve tubes, or they don't have the sieve plates. Therefore, this cannot be the xylem, and it has to be the phloem sieve tube elements. Therefore, the answer is going to be C. Question number 25. Which substance makes xylem vessels? Question number 25. Which substance makes xylem vessel walls impermeable to water? So you must know that it's always going to be lignin. The lignified walls makes the xylem waterproof. Question number 26. The following tissues carries an electrical impulse during the cardiac cycle. In which order does the electrical impulses travel during the cardiac cycle? So as we all know, it starts with the sinoatrial node, which goes through the muscles of the atria. So the sinoatrial node is at the right atrium, gives electrical impulses to the walls of the atria. Then what happens is that it meets the atrioventricular node, which delays the impulse, and then it spreads it first along the bundle of His and then to the Purkinje fibers for the ventricles to contract, and the atrioventricular node delays the impulse 0.1 seconds, so the atrium and the ventricles do not contract at the same time. So atrioventricular node, then Purkinje or Purkinje tissue finally. Therefore, the answer is going to be C. Question number 27. Which row is correct for the atrioventricular valve at P and for the semilunar valve at R? So we could see that this is ventricular systole. Now, what happens at ventricular systole is that now the ventricle contracts. Therefore, the pressure at the ventricle, so that's at the start only, the pressure in the ventricle is large enough. Therefore, what happens? It pushes the semilunar valve open and then forces blood outside, either into the aorta or the pulmonary artery. And then what happens is that the atrioventricular node shuts at the start. The reason for this is that the pressure in the atria is now very low and cannot overcome the pressure in the ventricles. Therefore, it pushes on the cuffs, shutting the atrioventricular valves at the start. Therefore, the atrioventricular valves at the start must be closed. Now, here it's at the end, so R, we could see it's at the end of ventricular systole, and we could see it's slightly merging at the start of diastole. Now, what happens in diastole is that the ventricle now relaxes and the pressure in the atria starts increasing. Now, if the ventricle relaxes, this means that the pressure in the ventricle is now really low, which is diastole. Now, what happens is that the pressure is no longer strong enough to push blood outside through the semilunar valve. Therefore, what happens is now the semilunar valve goes shut. Therefore, at R, the semilunar valve closes, and the answer is going to be A. Question number 28. Which conclusion is supported by the graph? Now, here we could see an oxygen dissociation curve for a human and for a cat. Now, for the cat, it's slightly shifted to the right. This means that the affinity of hemoglobin to oxygen is lower. Therefore, this means that hemoglobin of cats needs a higher partial pressure of oxygen to have the same saturation. Now, let's see the suggestions that we have here. A. The affinity of cat hemoglobin for oxygen is greater. Now, this is incorrect. It has a lower affinity because, as you can see, the graph is shifted to the right, and therefore the cat hemoglobin needs a higher partial pressure of oxygen in order to achieve the same oxygen saturation as for the human. Therefore, this is incorrect. For B. At high partial pressures of oxygen, cat hemoglobin picks up oxygen more easily. Now, this is incorrect. It picks up oxygen harder because, as we said, it has a lower affinity. Now, for C, at lower partial pressures of oxygen, oxygen is released more easily from cat hemoglobin than from human hemoglobin. Yes, it's released more easily because, as we said, it has a lower affinity. Therefore, this means it releases oxygen easier and binds to oxygen harder. For D, the shift in oxygen dissociation curve caused by Bohr effect is larger for cat hemoglobin than human hemoglobin. Now, Bohr effect is to do with the carbon dioxide concentration, not the percentage saturation. Therefore, this is totally incorrect, and the answer is going to be C. Question number 29. Which statement is correct about how oxygen combines with hemoglobin? So, when oxygen combines with hemoglobin, this forms oxyhemoglobin. And there's something called an allosteric effect. So as we all know that hemoglobin can bind 4 oxygen atoms. So as the first oxygen atom binds to hemoglobin, it makes it easier for the next and then the next and then the next oxygen atoms to bind to hemoglobin, causing an allosteric effect and an S shape in the graph. Now let's see the suggestions that we have here. Combining all 4 oxygen molecules with hemoglobin does not affect the shape of hemoglobin. Now, this is totally incorrect because something happens called an allosteric effect. So therefore this means it does actually end up affecting the shape of hemoglobin and changing the overall structure, therefore this is totally incorrect. For B, 1 oxygen molecule can combine with each heme group of the hemoglobin molecule. Now this is correct. Each heme group combines to 1 oxygen molecule. Be careful here of the wording, 1 oxygen molecule. And because we have 4 heme groups in a hemoglobin molecule, this means that it can bind to 4 oxygen molecules. Therefore, each heme group can combine 1 oxygen molecule, therefore this is correct. C, the first oxygen molecule to combine with hemoglobin does not affect the shape. Now this is incorrect because about the allosteric effect that we spoke about. So when the first oxygen molecule combines with hemoglobin, this makes it easier for the next oxygen molecules to bind, and this is called the allosteric effect, changing the overall shape. Therefore, these two must be incorrect. For D, the third oxygen molecule to combine with hemoglobin makes it more difficult. Now this is completely contrasts what we just said. It makes it more easier for the fourth oxygen molecule to combine, not more difficult. Therefore, the only correct answer must be B. Question number 30, how are alveoli adapted to their function? Now let's see the suggestions that we have here. A, they contain squamous epithelium for a short diffusion distance. Now this is correct. This makes diffusion faster, therefore gives the adaptation for the alveoli for more efficient gas exchange. Now for B, they contain globular cells. This is totally incorrect. They do not contain ciliated epithelium, and they do not contain cartilage. What contains all 3 of these are the trachea and the bronchi. Therefore, this is totally incorrect and the answer is going to be A. Question number 31, the diagram shows oxygen diffusing from the space inside the alveolus into the blood through the gaseous exchange surface. What would increase the rate of diffusion of oxygen from the alveolus to the blood? Now let's look at this diagram. Here we have oxygen concentration in the blood, and for X, oxygen concentration inside the alveolus. So the larger the gradient between these two, the more quicker the diffusion happens, because in order for diffusion to be faster, there has to be a more steep gradient. Therefore, what I want is either for the oxygen concentration inside the alveolus to decrease significantly and for the W to increase. This makes the gradient more steep, or the opposite, for this to decrease and this to increase, or for one of them to stay the same and one of them increases, for example. And now we have the thickness of the alveolar wall and the capillary endothelium. So, as you all know, rules of diffusion. So, the thinner the wall, the more faster diffusion would take place and the more efficient the gas exchange would be. Therefore, you want a thinner of alveolar wall and capillary endothelium. Now, let's see the suggestions that we have here. Here, what would increase the rate of diffusion of oxygen from the alveolus to the blood? So here's the alveolus to the blood. Now, what we want here is we know that for diffusion, it goes down concentration gradient. Therefore, I'm trying my best in this case to increase the oxygen concentration whilst decreasing the oxygen concentration in the blood, as well as decreasing the thickness, because the higher the concentration of oxygen here, the faster they move down concentration gradient, because this is how diffusion works. So let's see the suggestions that we have here. For A, increase in W and a decrease in X. This is not what we want. We want an actual increase in X. Therefore, this is incorrect. For B, an increase in W and a decrease in Y, and keep X the same. Now this is incorrect. We want an increase of X, not W. Therefore, these are incorrect. For C, there's an increase in Y. Now this is totally incorrect. I want to actually decrease the thickness of the alveolar wall. And then finally here, increase X which is what we want. We want the concentration here to increase to make a more steep gradient, so diffusion could happen faster into the blood. This is correct. And decrease Y, which is the thickness, which is correct. And we might end up keeping W the same as long as we focused on these two elements. Therefore, the answer is going to be D. Question number 32, which tissues are present in a bronchus? So, as I mentioned earlier, trachea and bronchus has both cartilage, cellulated epithelium, and smooth muscle. Therefore, the answer is going to be A. Question number 33, steep concentration gradients must be maintained for efficient gases exchange to occur in the human lungs, which correctly describes how steep concentration gradients can be maintained. So here we have elastic fibers in the wall of alveoli recoil when breathing out. Now this is correct. This does affect the concentration gradient because this recoiling creates a negative effect, helping to push air out. Therefore, this negative pressure does end up affecting the concentration gradient. Therefore, it ends up pushing air outside the lungs more efficiently. Therefore, this is correct. Now, continual supply of deoxygenated blood by the pulmonary artery. So, the deoxygenated blood creates an actual steep gradient whilst the air is oxygenated. Therefore, the less oxygenated the blood from the pulmonary artery and the more oxygenated the air, this means that it creates a more steep gradient. Therefore, this is correct and the answer is going to be A. Question number 34. What is the percentage decrease in the mean time that a person had diarrhea when they were treated with an antibiotic? So, this is the original value when the person was just treated with oral rehydration therapy versus when it was treated with an antibiotic. So, this is the mean time the person had diarrhea in days. So, in order to calculate any percentage decrease, what we do is we use a very basic formula. So, it's going to be 5.3 minus 3.2. So the difference between those values divided by the original value. So the original value that we have is going to be 5.3. Therefore, what we do is 5.3 minus 3.2. This is going to give us 2.1. And now we divide this answer by 5.3. This is going to give us 0.39. So in percentage, it's going to be A. Number 35. How does the antibiotic penicillin affect the metabolism of bacterial cell? So you should know that penicillin binds to transpeptidase, and therefore this prevents the formation of new cross-links. Therefore, this prevents the formation of bacterial cell wall. Now, let's see the suggestions that we have here. Penicillin reduces cell wall growth by preventing water uptake. Now, this is totally incorrect. Penicillin inhibits the formation of cross-links in the cell wall. This is correct, and the answer is going to be B. Question number 36. What is the percentage increase in multi-drug resistance infections between 2007 and 2015? So, as I mentioned earlier, any formula for percentage change is going to be as the following. So, we're going to have the difference. So 0.5 minus 2, so the difference between the two values divided by the original value. So because it rose from 0.2 to 1.5, therefore 0.2 is going to be the original value. And this is going to be the percentage change formula. So 1.5 minus 0.2 is going to give us 1.3. Now 1.3 divided by 0.2 is going to give us 6.5. Now, we want to multiply this value by 100, so 6.5 times by 100 is going to give us 650% change. Therefore, the answer is going to be D. Question number 37. Which method provides protection to infants against whooping cough and reduces the chance of developing this disease later? So, what provides future protection and reduces the chance of developing the disease is definitely going to be a vaccination with a primary immune response taking place. This is going to provide future protection for the disease because of what happens when the vaccine is provided to the infant. The antigen is acquired in the first time by the body. And then when the antigen is seen by the body, what happens is that now memory cells exist. And when the memory cells exist, if this antigen were encountered again in the future, now we have existing memory cells that would divide quicker by mitosis and then would release the antibodies in order to reduce the chance of developing the disease later. So here we're looking for the vaccinations. Therefore, any antibiotics is totally incorrect. Antibiotic is therapy when the person already caught the disease. It cannot provide any specific protection. First, C, injections of antibodies. Now, antibodies also is if the person actually got the disease and you're trying to provide post-protection from the disease. For example, the rabies vaccine provides antibodies when the person already might have suspected rabies. Now for D, injections of antigens from Bordetella pertussis bacteria. Yes, injections of antigens is technically a vaccine. Therefore, the answer is going to be D. Question number 38, the electron micrograph shows a type of blood cell. What can be concluded from the electron micrograph? So, we could clearly here see a large nucleus. Therefore, this means that transcription and translation take place here heavily. And we could see a lot of rough endoplasmic reticulum. Almost the entire cytoplasm is rough endoplasmic reticulum. Therefore, this shows us that this cell might be producing a lot of antibodies in this case. Now, we could kind of suspect that this cell might be a B cell or B lymphocyte because lymphocytes usually have a really large nucleus and they are circular in shape. Now let's see the suggestions that we have here. What can be concluded from the electron micrograph? A, the cell secretes products that are toxic to pathogens. So this is incorrect because products which are toxic to pathogens are cytokines. Therefore, this is incorrect. As we said, this looks like a B lymphocyte and B lymphocyte secretes antibodies. Now for B, the cell synthesizes a large quantity of proteins. Yes, this is correct because as we can see here, the rough endoplasmic reticulum, there's a lot, and the large nucleus gives an indication that a lot of transcription and translation takes place here. Therefore, a lot of protein synthesis takes place here to provide the antibodies. Therefore, this is totally correct. C, the cell synthesizes large quantities of antigens. Now, this is totally incorrect. Antigens are found outside, and you do not really need large quantities of antigens. Therefore, this is totally incorrect. D, the cell digests pathogenic bacteria. So, if we were to assume that this is a phagocyte, it would not look like this. First of all, phagocytes, such as a macrophage, have a kidney-shaped nucleus and has a lot of vacuoles in the cytoplasm and lysosomes in order to digest the bacteria. Therefore, it would look nothing like this blood cell that we see here. Therefore, this is totally incorrect, and the answer is going to be B. Number 39, why is mitosis important in the immune response? Now, what happens when an antigen is first encountered by immune cells? What happens is something called clonal selection takes place. And this is identifying which T helper cells are specific to the shape of the antigen that just entered, as well as the B cells which are specific to the shape. Now, what happens is that the lymphocytes which are specific to the shape of the antigen, what happens is clonal selection. Now, this specific shape, so the specific lymphocyte which is identical in shape to the antigen, now undergoes mitosis or division because I want more of these which are specific to the actual antigen, and this is called clonal expansion. Now let's see the suggestions that we have here. A, it allows B lymphocytes to produce plasma cells. Now this is mitosis, so this is correct. What happens is that B lymphocytes undergo mitosis to produce plasma cells and memory cells by the process called clonal expansion, so clonal selection of the B lymphocyte, which is identical to the antigen, and now clonal expansion, which is technically mitosis, is what takes place, forming plasma cells, which secretes antibodies and more memory cells. Now, for B, it allows the B lymphocytes to synthesize antibodies. Now, this comes after math, therefore this is incorrect. It's not directly related to mitosis. For C, it allows neutrophils to carry out phagocytosis. This is totally incorrect. Phagocytosis is more related to lysosomal digestion, not really related to mitosis, therefore this is incorrect. D, it allows C lymphocytes to recognize foreign antigens. Now, this is clonal selection, not expansion, therefore it has nothing to do with mitosis, and the answer is going to be A. Question number 40, what is fused with a B lymphocyte to form a hybridoma cells in monoclonal antibody production? So, how MABs or monoclonal antibodies are formed is using a B lymphocyte with a cancer cells. Now, these form a hybridoma. Cancer cells are also known as myeloma cells. Therefore, the answer is going to be D. Thank you very much for watching this video. If you have any more recommendations, leave them in the comment section down below. And please subscribe to this channel and like this video.